In the summer of 2013, inside a genetics laboratory at Stanford University, a team of researchers opened a set of results they had been waiting years to see. And what came back on their screens did not make sense at first. Blood samples had been collected from more than 1,000 people across Mexico, ordinary people from 20 different indigenous communities and 11 mixed mestizo populations. And when the data was run through the analysis, something appeared that should not have been possible according to everything scientists thought they understood about a single modern nation. Two groups of people, both living within the same country, both carrying Mexican passports, both raised under the same flag, showed genetic differences on the scale normally reserved for entire continents. The researchers checked the data again. They checked it a third time. The result did not change. A quiet, almost unbelievable alarm moved through the team, because if this was accurate, then the story every Mexican child had been taught in school, the clean story of one indigenous population meeting one wave of Spanish colonizers and blending into a single new people, was not simply incomplete. It was fundamentally wrong. And once that paper was finally published in the journal Science in June of 2014, it did not just add a footnote to Mexican history. It cracked open a genetic mystery nearly 10,000 years deep, one that is still being unraveled today, and one that touches more than 130 million people, who carry this ancient, layered inheritance in every cell of their bodies right now. For most of the 20th century, Mexico built its entire national identity around a single unifying idea. It was called mestizaje, the concept of the cosmic race, the belief that the true Mexican was something entirely new, a fusion born from the meeting of indigenous and European blood. Painted onto the walls of government buildings by muralists like Diego Rivera, taught in classrooms, recited by presidents, this was not just a demographic description. It was treated as a foundational truth, a source of pride for an entire nation trying to define itself after centuries of colonization and decades of revolution. But a national philosophy is not the same thing as a biological fact. And for generations, nobody actually had the scientific tools to check whether the poetry and the genome agreed with each other. The story taught in textbooks went like this. Indigenous peoples lived across the land for thousands of years. Spanish colonizers arrived in the 16th century. The two groups mixed, and from that mixing came the modern mestizo, the blended Mexican of today. It is tidy. It fits on a single page. And like most tidy stories about deep human history, it turns out to capture only a small fragment of something far stranger. That 2014 study, led by researchers from Stanford, the University of California, San Francisco, and Mexico's National Institute of Genomic Medicine, analyzed close to 1 million genetic variants pulled from that pool of more than 1,000 individuals. What emerged was not a gentle refinement of the old story. It was a demolition of it. The genetic distance separating some indigenous groups inside Mexico's own borders was not a small regional variation, the kind you might expect between neighboring towns or provinces. In some cases, it matched the genetic distance separating Europeans from people in East Asia. Consider two specific groups that researchers focused on. The Seri, also known as the Comcac, who have survived for centuries along the harsh desert coastline of the Gulf of California in the north, fishing and adapting to one of the most punishing environments on the continent. And the Lacandon, who live deep in the rainforest near the Guatemalan border in the south, in a world of humidity, dense jungle canopy, and an entirely different ecological rhythm. According to the genetic data, these two populations, both unmistakably Mexican, both indigenous to the same nation, are as different from one another as a person from Madrid is from a person from Beijing. Sit with that for a second. Not separated by oceans, not separated by different countries, separated only by mountains, deserts, and centuries of near total isolation from one another, and yet carrying genomes as distinct as those dividing opposite ends of the ancient world. This was not a difference of accent, of cuisine, or of local custom. This was a difference embedded in the deepest structural layer of human biology, the kind of divergence that takes many hundreds, often thousands of years of separation to produce. And the pattern did not stop with the Seri and the Lacandon. As researchers moved region by region across the country, they identified 3 distinct major genetic clusters, each one carrying the fingerprints of ancient civilizations, migrations, and adaptations that predate any European ship by thousands of years. In some communities, scientists could trace genetic echoes of long-vanished trade networks and ancient political alliances, threads still measurable in the DNA of people living hundreds of miles apart today. Why would a single country, roughly the size of Western Europe, contain this staggering degree of internal genetic diversity? The answer sits in geography and in time. Mexico is not a flat, easily crossed landscape. It is carved lengthwise by 2 enormous mountain ranges, the Sierra Madre Occidental and the Sierra Madre Oriental, and it contains high central plateaus, dense southern rainforest, and vast northern desert, often within a few hundred miles of each other. For thousands of years before any European vessel appeared on the horizon, these natural barriers kept populations apart, forcing them to adapt independently to wildly different environments and to slowly accumulate their own distinct genetic signatures, generation after generation. Picture a farming community tucked into a fertile valley in Oaxaca, cut off by a wall of mountains from a fishing community on the Pacific coast, only 100 miles away as the crow flies. For centuries, maybe millennia, those two communities might trade occasionally and share the odd traveler, but they would overwhelmingly marry within their own populations and pass down their own particular genetic inheritance across dozens of generations. Multiply that pattern across dozens of valleys, coastlines, deserts, and highland plateaus, sustained not for 1 or 2 centuries, but for thousands of years, and you start to understand exactly how one modern nation ended up harboring this much internal genetic variation. This is the piece the simple textbook story leaves out entirely. Mexico was never a single unified indigenous population sitting passively and waiting for European contact. It was, and in many ways still is, a mosaic of dozens of distinct peoples, each carrying their own deep independent history stretching back long before Columbus was ever born. The genetic diversity researchers are mapping today as a direct, measurable record of that reality, written not in stone monuments, but in the molecules of living people. But the discoveries did not stop at indigenous diversity. If that first finding reshaped how scientists understood Mexico's ancient past, the next major line of research reshaped how they understood its colonial past. And it did so by focusing on one extraordinarily powerful genetic thread, the DNA passed down exclusively through mothers. This is called mitochondrial DNA, and it carries a unique property that makes it invaluable to historians and geneticists alike. It is passed down almost unchanged from mother to child across generations, preserving an unbroken maternal lineage, stretching back thousands of years, essentially untouched by the paternal line at all. When researchers analyzed mitochondrial DNA from more than 2,000 samples collected across the country, the results were staggering. Close to 90% belong to the 4 major indigenous American genetic lineages found throughout the Western Hemisphere. 90% in a country where the majority of the population identifies not as indigenous, but as mestizo, mixed, and whose modern national identity has for a century been framed around the fusion of European and indigenous roots in supposedly equal measure. The maternal genetic record tells an overwhelmingly different story, one dominated almost entirely by indigenous ancestry, carried forward through the mothers of the nation. This finding forces an uncomfortable but historically important reckoning. It suggests that the mixing that defined the colonial period was never a story of equal partnership between two peoples meeting on level ground. It was overwhelmingly a pattern in which European men fathered children with indigenous women, while the reverse, European women having children with indigenous men, was far rarer. Historians studying the demographics of Spanish colonization already suspected this, since the overwhelming majority of arrivals from Europe during the early colonial decades were male, often soldiers, administrators, and merchants traveling without wives or families. But to see that hypothesis confirmed with this degree of biological clarity, written directly into the cells of more than 100 million living people, is something else entirely. It transforms a historical suspicion into hard, measurable, undeniable data. And this single detail changes how we should think about mestizo identity altogether. It is not a clean 50-50 blend, a neat average between 2 ancestral populations sitting side by side. It is an asymmetric inheritance, an identity built disproportionately, at least along the maternal line, on indigenous foundations. Even while Mexican cultural identity has often been framed around its European inheritance, the surnames, the language, the colonial architecture, the DNA remembers what the culture sometimes chose to forget, a quiet and persistent record passed mother to daughter, across 5 centuries, waiting for the technology to finally catch up and read it properly. There's a third thread woven into this story too, one that has been left out of the national conversation for far too long. Beginning in the 16th century, tens of thousands of enslaved Africans were forcibly transported to the shores of New Spain, and their descendants became and remain a real and measurable part of the modern Mexican gene pool. For centuries, this African ancestry was quietly erased from the national narrative, overshadowed by the simpler, more convenient story of a purely indigenous and European mixture. But the genetic record does not forget or negotiate. Contemporary genome studies consistently detect African ancestry components, sometimes modest, sometimes substantial, threaded through populations along the Gulf Coast and the Pacific Coast in particular, precisely the regions where the historical footprint of the transatlantic slave trade runs deepest. This is not a footnote buried at the bottom of a research paper. It is a third foundational pillar of the modern Mexican genome, standing alongside the indigenous and European contributions. And researchers are only now beginning to give it the sustained attention it has always deserved. Now push the timeline back even further, past the colonial period entirely, past the height of the Aztec Empire, past even the flourishing of Maya civilization, to a moment nearly 10,000 years in the past, when the ancestors of Mexico's indigenous peoples were still shaping the very foundations of their distinct genetic identities. In 2021, a team led by researchers at Mexico's National Institute of Genomic Medicine published a genome-wide analysis of 716 individuals drawn from 60 of the 68 officially recognized ethnic groups across the country. The goal was ambitious, almost to the point of audacity, to reconstruct the deep demographic history of indigenous Mexico using nothing but the silent testimony locked inside human DNA, since almost no written record from that era survives. What they found was a genetic splitting point between two major ancestral population groups, one associated with the harsher, more arid regions of northern and central Mexico, known to archaeologists as Arido-America, and the other associated with Mesoamerica, the fertile heartland where complex civilizations would eventually rise. This split occurred somewhere between roughly 4,000 and nearly 10,000 years ago, and the timing is not a coincidence. It lines up almost precisely with one of the most consequential developments in human history anywhere on the planet, the beginning of sedentary agriculture in Mesoamerica. Think about what that actually means. As ancient peoples in what is now central and southern Mexico began transitioning away from nomadic hunting and gathering towards settled farming, cultivating maize, beans, and squash, the very crops that would one day feed entire empires, their populations began to diverge genetically from their more mobile, arid-dwelling neighbors to the north. Agriculture did not simply change how people ate or where they slept. It changed the very architecture of their family trees, locking in genetic differences that scientists can still detect and still measure nearly 10 millennia later. This finding pushes the true origin story of the Mexican people back far beyond anything most people were ever taught. It is not a story that begins with Hernán Cortés landing on the coast near Veracruz in 1519. It is not even a story that begins with the rise of the Aztec Empire in the 14th century or the flourishing of Maya civilization centuries earlier. It is a story that begins with the earliest farmers of Mesoamerica planting the first domesticated crops in the Americas, setting into motion a genetic divergence that echoes across nearly 10,000 years, all the way into the DNA of Mexicans alive right now. That deep timeline connects to an even larger, continent-spanning story, the peopling of the Americas itself. The ancestors of Mexico's indigenous populations were part of the broader human migration that first crossed into the Western Hemisphere many thousands of years earlier, spreading gradually southward and adapting to an extraordinary range of environments, from Arctic tundra to tropical rainforest to high-altitude plateau. Some of these earliest migrants likely moved along ancient coastlines, following marine resources southward across many generations, while others pushed inland through plains and river valleys, eventually reaching the rugged terrain that would become central Mexico. Each wave of movement, each new environment encountered, left its own subtle imprint on the genome, an imprint modern researchers can still detect, date, and map onto a physical landscape. By the time agriculture began transforming life in Mesoamerica, these populations already carried thousands of years of independent history, independent adaptation, and independent genetic development behind them. With this deep ancestral picture established, researchers turned to a different but equally urgent question. What does all of this mean for the health, the medicine, and the future of the people carrying this extraordinary genetic inheritance today? One of the most ambitious efforts to answer that question came from the Mexico City Perspective Study, a massive undertaking that genotyped and sequenced more than 140,000 adults, results of which were published in the journal Nature in 2023. This is not a modest sample size. It stands as one of the largest genetic studies ever conducted on a Latin American population. And its findings are actively reshaping how medicine approaches genetic risk in Mexican and broader Latino communities worldwide. The study confirmed what earlier research had suggested, that most individuals carry a complex blend of indigenous American, European, and African ancestry, with particularly extensive indigenous contributions concentrated in the central, southern, and southeastern regions of the country. But it also uncovered something with direct, immediate medical significance. Segments of the genome tracing back to indigenous Mexican ancestry showed a distinct pattern: lower overall levels of certain coding variation, but a notable excess of what geneticists call homozygous loss of function variants, essentially genetic changes where both copies of a gene are altered in ways that reduce or eliminate its normal function entirely. Why does that matter? Because these kinds of variants can have outsized effects on health, on how the body metabolizes certain medications, on susceptibility to specific diseases, and on a wide range of biological processes. Doctors are only beginning to map in detail for indigenous Mexican populations specifically. For decades, the overwhelming majority of genetic research and genetic reference databases used in modern medicine were built almost entirely from populations of European ancestry. That means an enormous, medically important slice of human genetic diversity, the slice carried by indigenous Mexican and broader Latin American populations, was essentially invisible to precision medicine for generations. This new wave of research is beginning to close that gap, one variant, one gene at a time. One of the most recent chapters in this unfolding story comes from an ambitious effort known as the Mexican Biobank, which analyzed genetic data from more than 6,000 individuals across the country and produced something genuinely new, a publicly accessible resource designed specifically to support ancestry-informed genetic testing for Mexican and broader Hispanic populations. What this research revealed was striking. Even within Mexico, allele frequencies, essentially how common certain genetic variants are in a population, differ dramatically from region to region, shaped both by overall admixture proportions and by which specific local indigenous ancestral group contributes most heavily to a person's genome. Someone with substantial ancestry linked to central Mexico may carry a meaningfully different genetic risk profile than someone with predominantly southern Zapotec ancestry or Maya ancestry from the Yucatan Peninsula. These are not small academic footnotes. They carry direct, real-world clinical consequences. Researchers have identified specific variants with clear medical implications, including one that affects how the body metabolizes fentanyl, a powerful pain medication with particular relevance for post-surgical pain management and safer opioid dosing during childbirth, and another that affects statin metabolism, relevant to the cholesterol-lowering medications taken by millions of people worldwide to reduce cardiovascular risk. Without an ancestry-informed, regionally aware approach to genetic testing, these kinds of variants can be entirely missed, leading to medication doses that are either ineffective or dangerously miscalibrated for a given patient. A dosage calculated using a generic, one-size-fits-all genetic model built primarily on European reference data might work perfectly well for one Mexican patient and fail entirely for another, simply because of which region and which indigenous ancestral group their family happens to trace back to. Some of the clearest examples of why this matters emerged even before these newer, larger studies were completed. Years earlier, researchers studying the genetics of Mexican and Latino populations identified a variant in a gene called SLC16A11, strongly associated with increased risk of type 2 diabetes. It turned out to be far more common in populations with substantial indigenous American ancestry than in populations of primarily European or Asian descent, a finding that likely never would have surfaced if researchers had continued relying almost exclusively on European ancestry datasets. Around the same period, studies following Mexican and Mexican-American children found meaningful differences in lung function and asthma outcomes that tracked closely with the proportion of indigenous versus European ancestry in a child's genome, alongside differing rates of breast cancer and differing responses to certain treatments across ancestry groups. These were not small, abstract statistical footnotes. They were real, measurable differences in real people's health outcomes, hidden in plain sight for years, simply because nobody had built the genetic tools capable of seeing them clearly. This is where the deep ancestral story and the modern medical story finally converge. The same forces that produced Mexico's extraordinary genetic diversity, thousands of years of geographic isolation, independent adaptation across dozens of distinct populations, and a colonial period that reshaped the genetic landscape all over again, are the very same forces that now demand a more sophisticated, more regionally aware approach to medicine. You cannot treat a genetically diverse population as though it were genetically uniform. The data simply will not allow it. And this raises a broader point worth sitting with. For most of the history of modern genetics, Latino populations, including Mexican populations specifically, have been dramatically underrepresented in scientific research, despite making up close to 10% of the world's population. Hispanic and Latino individuals have historically accounted for less than 1% of participants in major genetic studies, while European populations, making up less than 20% of humanity, have accounted for the overwhelming majority of research subjects. This imbalance was never a reflection of importance, and it was certainly never a reflection of scientific curiosity. It was a reflection of where research funding, infrastructure, and institutional attention happened to be concentrated for the better part of a century. The consequence was a massive, medically consequential blind spot, one that studies like the Mexico City Perspective Study and the Mexican Biobank are only now beginning to correct. So what does all of this add up to? What is the real, complete answer to the question of where Mexicans actually come from? It is not a single simple answer, and it never could be. It is a layered answer built across nearly 10,000 years. It begins with ancient populations spreading across an entire hemisphere and adapting over thousands of years to environments ranging from harsh northern desert to lush southern rainforest. It continues through a pivotal agricultural revolution roughly 4 to 10,000 years ago, one that split ancestral populations into distinct genetic lineages still detectable today. It moves through the rise and fall of extraordinary civilizations, the Olmec, often called the mother culture of Mesoamerica, whose massive carved stone heads still stare out from the jungles of the Gulf Coast, the Maya, whose astronomers tracked the movements of the stars with an accuracy that still impresses modern scientists, the Zapotec, who built one of the first true cities in the Americas atop a leveled mountain in Oaxaca, and finally the Mexica, the people commonly called the Aztecs, who built a lake-bound capital so advanced that the invading Spanish compared it to the great cities of Europe. Each of these civilizations rose, transformed its landscape, and eventually fell, but none of them vanished without a trace. Each left behind not just monuments and writing systems, but genetic fingerprints still traceable in modern populations today. And then the story passes through a colonial period whose true demographic character, an asymmetric mixing dominated overwhelmingly by indigenous maternal lineages, is only now being fully understood through the lens of modern genetic science. The result is not a simple blend. It is not a straightforward, even 50-50 mixture of two ancestral groups, combining neatly into a single homogeneous whole. It is something far more intricate, a genetic mosaic reflecting one of the most complex, most regionally diverse demographic histories found anywhere on the planet. Some indigenous groups within modern Mexico remain as genetically distinct from one another as separate continents. Maternal lineages remain overwhelmingly indigenous, even in populations that culturally identify primarily with their European heritage. And regional genetic variation continues to carry real, measurable consequences for modern medicine, for how doctors prescribe pain medication, for how they manage cardiovascular risk, for how precision medicine itself must evolve to serve this population accurately and safely. None of this diminishes Mexican identity. If anything, it deepens it, roots it further back in time, and reveals it to be far richer and more complex than any single simplified narrative could ever capture. This is not a story about Mexicans being somehow different from who they always believed themselves to be. It is a story about how much deeper, older, and more intricate that identity truly is once you look past the surface and into the biological record itself. What the science ultimately reveals is resilience, populations that survived isolation across some of the most demanding environments on Earth, populations that endured a demographic catastrophe during the colonial period so severe that entire lineages were reshaped, and yet an overwhelming indigenous genetic legacy was still preserved, passed down through generation after generation of mothers. Populations that today are finally being studied with the scientific rigor and the genetic detail they have always deserved, closing a gap in medical knowledge that should have been closed decades ago. This is not a story with a single origin point, a single ancestor, or a single moment of creation. It is a story written across 10,000 years, across mountain ranges and deserts and rainforests, across empires and conquests and quiet centuries of survival, and it continues to be written today, one genome, one study, one discovery at a time. The next time someone tries to explain the story of the Mexican people in a single sentence, remember what the data actually says. Remember the Seri and the Lacandon, as different from each other genetically as Europe is from China. Remember the 90% of maternal lineages tracing back to indigenous roots. Remember the agricultural revolution, nearly 10,000 years old, still visible in the genome of every person carrying that ancient inheritance forward. Because the real story was never simple, and it was never meant to be. Simple stories are convenient. They fit neatly into a single paragraph, a single mural, a single chapter of a school textbook. But convenience was never the same thing as truth, and for the first time in history, scientists finally have the tools to tell the difference. Every strand of DNA, carried by every person of Mexican descent, whether they live in Mexico City or Los Angeles, in a small village in Oaxaca, or a suburb in Texas, is itself a kind of historical document, a record written not in ink, but in molecules. One that has survived conquest, survived colonization, survived centuries of upheaval, and arrived intact in the present day. Scientists are only just learning to read that document properly, and every new study adds another page to a history far older, far richer, and far more resilient than any single textbook sentence could ever hope to capture.